All right, let's jump right into one of the most incredible, most fundamental processes in all of biology, DNA replication. We're going to break down how a single, tiny cell pulls off a copy job that is so massive and so incredibly precise, it's honestly hard to believe. No, really. Just stop and think about that for a second. We're talking about organisms like E. coli, right? Things smaller than a speck of dust. And they're running this molecular machine with just mind-blowing speed and accuracy.
So, what are we talking about here? We're talking about 4.6 million base pairs. That's 4.6 million individual letters in the genetic code, all packed into a single E. coli chromosome. I mean, that is literally an entire instruction manual for how to build and run a living thing.
And here's the part that just blows my mind. The cell copies all 4.6 million of those letters in about 42 minutes. How in the world is that even possible? Well, that's the secret, right? The key to understanding this whole thing is to stop thinking about it as some random, chaotic process. Instead, picture a highly organized, super efficient, and perfectly choreographed molecular assembly line. So, first things first, this whole operation isn't just starting at one end and chugging along to the other. No, it's way smarter than that. It starts right in the middle, at a specific origin point, and then it opens up in two directions at once. It's just like unzipping a jacket from the center. This one little trick instantly doubles the speed right out of the gate. So for Act 1, setting up the factory floor. You can't just start building. You need a blueprint, you need to mark the foundation, and you need to get all the heavy machinery in place. That's exactly what the cell does first. And this process doesn't just start anywhere. The assembly line has a very specific starting point. For E. coli, it's a 245 base pair sequence called ORI-C. Think of it like a giant neon sign on the chromosome that flashes, "Start here." And it's no accident this spot was chosen. It's packed with adenine and thymine bases, A's and T's, which are kind of weakly linked together. This makes it the perfect, easy-to-open spot to get the whole process started. And here's how it goes down. A team of proteins called DNA, let's call them the site foreman, shows up. They recognize and grab onto that ORI-C start site. This binding actually bends the DNA, putting stress on it, kind of like twisting a rope. And that tension is just enough to pop open that weak AT-rich section, creating the perfect little bubble for the real work to begin. Okay, so just to quickly recap the setup phase. The foreman, DNA A, binds and bends the DNA. This pops open a bubble at that weak spot. And then the heavy machinery arrives. A crucial enzyme called DNA helicase, the master unzipper, gets loaded onto each of the two separated strands, ready to tear off in opposite directions. This whole operation is a team effort, and it's time to meet the key players on our assembly line. We've just met helicase, the unzipper, but there's also primase, who acts like the ignition switch. Then you've got the star of the show, DNA polymerase III, our master builder. Following behind is DNA polymerase I, the cleanup crew. And last but not least, ligase, the final welder who makes sure every last connection is solid. Okay, the factory is set up, the crew is on site, it's time for Act 2, the dual assembly lines. This is where the magic really happens. Let's get building. And I just want to pause for a second to appreciate the sheer speed we're talking about. That master builder, DNA polymerase III, it's adding 1,000 new letters, 1,000 nucleotides every single second. That is just unbelievably fast. But there's a problem, a big one. The two strands of DNA are anti-parallel, which is just a fancy way of saying they run in opposite directions. It's like a two-lane highway. The problem is, our master builder, DNA polymerase, can only travel in one direction. It can only add new pieces to one specific end of the DNA chain. It's a one-way construction vehicle. So what do? That's the big puzzle. If your main builder can only move in one direction, how on earth do you copy the strand that's running the other way, especially while the whole thing is unzipping? Well, the cell's solution is absolutely brilliant. It runs two totally different assembly lines at the same time. For the one strand, the leading strand, it's easy. The builder just cruises along continuously right behind the unzipper. But for the other one, the lagging strand, it has to get creative. It builds backwards in a bunch of short little fragments. And you can see exactly how this works right here. The bottom strand, that's our leading strand, see? It's just one smooth, continuous process. But look at the top one, the lagging strand. It's a whole different story.
The polymerase has to wait for a bit of DNA to unzip. Then it hops on, builds a short piece backwards, away from the fork, then hops off and does it all over again a little further down the line. These short little pieces that get made on the lagging strand actually have a name. They're called Okazaki fragments. Each one is like a little mini segment of DNA that, as you can probably guess, is going to have to be stitched together later on to make a complete strand. All right, we are in the home stretch. The copying is almost done. But, a good factory doesn't just throw things off the assembly line. We're now in Act 3, quality control and shipping. It's time to clean up, proofread, and get these new DNA molecules ready to go. Now, because E. coli has a circular chromosome, our two replication forks have basically been in a race around the track, heading right for each other. To make sure they don't just crash, there's a specific terminus region. Think of it like a molecular finish line with a one-way gate. A protein called TUS sits there and essentially traps the forks, making sure the whole process stops cleanly and doesn't run wild. Okay, now it's time for cleanup. Remember those little RNA primers that we used to get things started? Well, they were just temporary. Our cleanup crew, another enzyme called DNA polymerase I, comes through, rips out those primers, and fills in the gaps with the proper DNA. But that still leaves a tiny little nick in the backbone. That's where our final welder, DNA ligase, comes in and seals everything up, creating a perfect, unbroken strand. It's a super important quality check.
The job is pretty much done, but there's one last, kind of weird problem. Because we copied a circle, we now have two new circles. But they're often interlocked, like rings in a magic trick. So for the final shipping step, an enzyme called topoisomerase performs a little bit of molecular magic. It snips one of the rings, passes the other one through the gap, and then seals it back up. Problem solved.
And there you have it, the end result. From one original DNA molecule, our incredible molecular factory has produced two absolutely identical copies. The factory's work is done. It has doubled its product, and now each of these DNA molecules is ready to be passed on to a brand new daughter cell. I mean, it is just an astonishing feat of molecular engineering. This tiny microscopic factory has a near-perfect accuracy rate. So what does that precision actually mean? It means everything. This process happening countless times every single second all over the planet is what ensures life goes on. That accuracy is the bedrock of heredity, of evolution. It's the foundation for the continuity of life itself.